Chris Moore is professor of neuroscience at Brown University and associate director of the Carney Institute for Brain Science. He is formerly associate professor of neuroscience at MIT, where he earned his PhD. His work has earned honors from the NIH and institutes at Harvard, MIT, Amherst, Dartmouth, and others. At the Carney Center, his team is helping to create tools for controlling circuits and image activity in the brain, using novel methods for transmission of optical signals between synapses. A major focus is dynamics in non-neural systems, referring to millisecond to second timescale changes that impact behavior in real time. Most notably, the research proposes a framework for vascular contributions to information processing in the brain. Chris has a high commitment to teaching inquisitive students and to applying findings from research to practical issues that we all confront in everyday life. So welcome, Chris, and take it over.
Thanks, Tom. And thanks a lot for having me. I very much appreciate it. And it's wonderful to see Maynard again, who I've had a lot of great interactions with over the years. I'm really interested in maybe in the question and answer is the best way, of course, because it should be a conversation. I'm deeply interested in talking about connections of what we're doing more directly to issues of different states of awareness and potentially religion. So if people want to talk about that, that'd be great. So let me share my slides here. So imagine that you are sitting on your porch, wherever your porch might be. This porch happens to be in Vermont, right on the Canadian border. In fact, you're looking at Canada right now. Well, the picture is looking at Canada right now. And just for a second, imagine how it smells. Or imagine the sound it's making. Or imagine the place in your life that you have ever sat that was most like this place. And I don't think it's too much to say, given the group we have. To me, this is a pretty exalted picture. It actually is a bit spiritual. Maybe imagine what your spiritual sense is vibing to you about this. Maybe that's too hippie of a way to put it, but it really is a picture that has more dimensionality. So the thing I'm trying to get at by asking you to just do all of those imaginations for a moment is just how complicated even the most simple scene we look at is. Imagine who's the person you're most likely to be sitting next to on the porch, or the person you'd most like to sit next to, which could be different than the person you're most likely to. So if you take a second and actually let your mind go there, it's really amazing the complexity that a single simple moment of vision can have and all the different rich associations it can make. So this is one way of entering the question of how the brain works. But I say that with an asterisk that we will take away by the end of this conversation, not just how the brain works, but maybe how the mind works is a better point. And what I propose we discuss at first here is what might be called a Turing test for a brain computer, which is somehow all those things you did were done by the biological computers that are contained within you. Now, how is it the case that using cells and using neurons in your brain, and we'll talk about what neurons are more later, are able to come up with the complexity of the imagination of the person likely to be sitting with you if you were sitting in a place that's like the one in the picture that was showing the rain in the scene from Vermont? That's actually amazingly complicated. So if we start to think about looking at the body and imagining in a bit as if we're from outer space, and we don't already have the bias that a bunch of neurons in the brain are the sole drivers of how we become who we are, which I imagine is an imagination that's friendly to this group's purpose. If we're imagining that, what would the requirements have to be? What would we have to see to believe that a system in the body is playing a role in creating our ability to appreciate, imagine about, remember, make inferences from a complex scene like the one I just showed you? So one of the features that I think would be pretty straightforward, I think everybody agrees the brain has something to do with this. And there's pretty widespread agreement, although there's lots of debates about the details in my field. But if you look at a brain, this is a colorized version of the human brain, there's pretty widespread agreement that throughout the brain, distinct areas of the brain do specific tasks. So for example, if you look right here, if you see that sort of fuchsia colored squiggle I'm moving my cursor on right now, that's a part of the brain that I study a great deal. It's called somatosensory cortex. But right, that somatosensory cortex is actually the part of what's called the neocortex. I'll explain those terms later. But it's the part that represents your body senses. So exactly this spot where my cursor is right now is where in almost everybody's brain the representation of feeling things with your fingers is. And if you go a little bit down from it, your lips, feeling with your lips is somewhere around here, right next to the fingers, so there's sort of a map of the body laid out there. And if I was able to stimulate your brain, you would actually feel tingling on your fingers if I stimulated here, and your lips if I stimulated there. Whereas, for example, more frontal regions have very specific functions in how we understand sequences. This is something scientists at Brown are working on. Here in this part of the brain, there are really interesting brain areas that represent faces, the visual image of a face. So if you're looking at my face right now in the Zoom call, I'm activating a part of your brain down there. So activity that is involved in creating rich behavior, the kind of really complicated behavior that our brains can do, that activity should predict the brain locations known to do those tasks. So we know this is the body representation here in that purple. We know that from 100 years of study, even more than that. So if we want to explain the ability to perceive with your hand, it probably has to do with something dynamic in some way happening in that area. Now, another important requirement is that if we believe some process in our body is playing a role as the computer that would let us do things like pretend to smell the rain off of a porch, it has to be at least as fast as the phenomenon of smelling the rain off the porch. In other words, it has to at least act as quickly as you can imagine it, which sounds like an obvious constraint, but it's actually a really useful one when we start to think about looking at different body systems and how they might contribute to understanding. Third, it has to have enough precision to explain the phenomenon. So right now, let's think for a second. And don't think about the picture that I just offered to you. Instead, think about some other porch that you have ever been on. It could be a stoop, a porch, something like that. Just imagine it for a moment. Now try to think of as many details that you can see or that you remember about that porch. All right. Any mechanism in the brain or the body together that would support your ability to have those imaginations has to have, and this is something that I guess will resonate with Don, sort of encoding depth. It's got to be able to be precise enough that I was imagining the stoop of the house I grew up on. It was a brick house. It was a brick facade with cement stairs, and there was actually a cement walkway my grandfather made. In imagining that, and then I got to write my initials in it, which was a great detail at the time, very important to me as a kid. That's a lot of details, and they're coming flooding back into my memory. I have to have a system that's got enough complexity that could represent all those details and many more. It's got to have an ability to have precision in its activity. So not only fast, but you could imagine something that's fast but doesn't have a lot of flexibility. And then you could imagine something that's got a lot of detail but can't move very fast. These are requirements if it is a kind of system in the body that's going to support things like ongoing emergence of perception, action, things we'll want to do, things we imagine, things we remember. And a final requirement, this isn't by any means a complete list, is that whatever system we find that can have this precision, move this fast, is in the right place at the right time to do it, if you will, that it has an ability to talk to other networks. We know that the brain and parts of the brain don't act alone. For example, the body map right here heavily interconnects with the motor map right here. How we perceive stuff tells us a lot about how we should move our hands, for example. So this Turing test, I should have explained, you may have heard of the Turing test. It's commonly known, although this wasn't the original way it was proposed, it's commonly known as a question of whether or not a computer, if you couldn't see behind a curtain and a computer was providing all the answers, could it fool you into thinking it was a person? Many people feel the Turing test has been solved by modern AI. I would agree with them. I definitely could be fooled that I was interacting with an AI, but that's a matter of debate. So what I'm asking about now is, could a system, if we just listed its properties, convince us that it was an information processing system? All right, so this is the test that we'll be thinking about as we think about our questions today. Now, the problem that we want to solve, as I've motivated, was a problem of the complexity of us looking at a picture, me speaking the English language, and you having complex thoughts that reflect that interaction. Now, the problem we evolved to solve, though, was this one. Imagine you're an early mammal or little mouse, beautiful, cute little mouse here. You've got to decide if you're willing to leave your burrow to go get a drink of water. Now, for us, that's not an existential choice. We can go to the water fountain. Maybe some of you have a drink next to you in your water bottles or glasses or something. But for a mouse, that choice is existential. There are a lot of different creatures that like to eat mice, that in fact, it's their primary meal. Every time it leaves its home, it's got to make a complex decision, which is, are its needs appropriate to the actions it's going to take? Because if it gets that wrong, it could be risking its life, or it is risking its life. It's got to decide whether it's worth risking its life for. Now, how is it that these early mammals that weren't overburdened with looking at pictures of porches, how is it that these early mammals actually survived when they don't have talons or beaks or snake skin or fangs? Well, there's a new invention that came about in the brain with mammals, and that's an invention called the neocortex, neo for new. So this new cortex is not something at all present, for example, in the brain of a frog. And in the brains of reptiles and birds, there's kind of a proto version of it, but it's not really the version mammals have. Mammals have a specialized version of it. And not only is it a distinct thing that mammals have, it's more or less the number one thing that's changed about the mammalian brain through mammalian evolution. So that mouse that I showed you has a neocortex, and almost the exact same neocortex it has, we have as well. We just have a lot more of it. Estimates are usually that we have 10,000 times more than a mouse, but not because we made the cortex different, not because we made the neocortex larger. It's the same, actually, here's a diagram of it. It's the same 6-layered circuit in us that it is in a mouse. So in my laboratory, we've taken data from mice and actually used it to model the human brain. And it's amazing how well you can do modeling parts of the human brain using a mouse brain. You get the basics right. So there's something very powerful about this cortical circuit that allows us to survive despite not having the physical attributes of things like snakes and dinosaurs and all these nasty predators with fangs and claws and things. And the neocortex is one of the big reasons why. Now, that neocortex needs a lot of training, just like AI does today. So it makes sense that we would keep our young at home for a long time, because what it basically does is a neocortex, well, I should say what it doesn't first. So snakes have an amazing sense of perception. Owls have the ability to hear mice and know exactly where they are at a distance in the dark with unbelievable precision. They can then fly, which is an incredible act of motor coordination with precision, and go get that mouse. You don't need a neocortex to perceive or to act, to make actions, to fly. What you need a neocortex for, there's increasing agreement on, is you need it for the ability to take a bunch of different abstract variables in the world and combine them so that you can act in real time on them. So I can do things like say, people that I have never met before, or some of them I've never met before, please imagine what it's like to look off of your porch. That act of using language, that's something I had to learn in my lifetime, but in real time, I can come up with a combination of variables that lets me choose the right word. That's the kind of thing the neocortex can do. Now, it does it through action potentials in part. Now, what action potentials are a way of two neurons communicating with each other. If you have heard of neurons, you probably heard that they're the cells that do the thinking in the brain. If you haven't heard of them, what they are is cells that have long processes that come off of them, known as axons, and other long processes that come off of them as dendrites. They're kind of like a ball on two strings, or they look more like a triangle often with two strings. The dendrites are good at listening to what other cells have to say in the brain. And then it goes through a big cell body and then goes out the axon if it wants to relay a signal to a downstream cell. And that's how one of the main ways that we do computation in the brain. It's the main way that the brain talks to itself is sending signals down these axons. Now, these signals are shown here in this movie. It's an electrical voltage event that actually goes down that long, thin bit of membrane. It causes calcium, same kind of calcium that we have in vitamins every day, to come into the ending of the cell. And then one neuron will release chemicals to talk to its neighbor, the next neuron over. As I'm showing in this movie, starting again now. So there's an electrical event that travels down that axon. A bunch of calcium comes in, and then a neurotransmitter chemical is released. And then the next cell hears that signal and will decide whether or not to pass it on. That sort of interaction between neurons is inarguably a big part of how we do computation in the brain. There's the chemical signal. And it looks like this. So what I'm showing you on the right side of the screen, I apologize, that's a little bit dim looking, is a movie that my laboratory took of what it looks like for a mouse to think. So every flashing light you see there, or part of how it looks for a mouse to think, every flashing light you see there is one neuron being active and firing an action potential. We're using a technique where we turn actually calcium events in cells into flashes of light. That way we can make movies of it and try to look and understand the patterns so we can know whether or not that's the part of the brain doing the information processing. And the whole problem is, the whole question is, for any given set of cells, they're being active at a given time. I put some triangles there just to capture different cells that are being active. Which ones actually matter to help us take the best action at a complicated moment, like choosing whether or not to go get a drink if you're a mouse and you're scared that the owl is going to eat you? It's how do we pick out, of all the billions of neurons we have, the right group that lets us go on and make decisions, take actions, learn from those decisions. Now, I just gave you the standard take on neuroscience in the world. So there are many, many more brain areas than just the neocortex. I happen to study the neocortex mostly. We study some other brain areas too. But that's the canonical view. That's the dogma, that there's a bunch of neurons in networks. And what they do is they flicker at different times. And if the pattern's just right, you'll have a thought like, those look like raindrops, or I bet it smells like porch protecting solvent. Because I can tell they had to weatherproof that porch. Now, Aristotle, interestingly, had the same question we do that we're trying to solve right now, which is, what are the biological computers within us? And Aristotle knew some really useful facts. So Aristotle knew that when people got injured in the head, they wouldn't think as clearly, they might have trouble moving, that the command center had to be in your head. This is, 300 some odd BC, so it's pretty good inference. He also knew that to store information, you probably had to have a complex network of interacting terms, just like our Turing test. And so he did experiments. In contrast to Plato, who's shown in the red robes here, Aristotle, shown in the blue robes, isn't just pointing up to the sky and saying, oh, I have a great idea, that's good enough. He was big on gathering data. So he went out and gathered data looking for a network in the brain, and he found one. He found blood vessels. He looked in, I believe it was 33 different kinds of creatures, and in all of them that had established brains, he found that there was a beautiful network in exactly the right spot, that he knew there had to be activity ongoing to create things like complex thoughts, because when people got injured there, the complexity of their thoughts sometimes went down. He found the vasculature. He found the blood vessels. This is actually, for those of you who are like to follow philosophy, Aristotle is often thought of as having gotten it wrong in thinking about where we come from as humans, as individuals, because he used to say that the heart was the seat of the soul. It was actually a really good inference that he made. He went looking for a network in the head, in about the position of the brain. He found one. He found one that looked a lot like the kind of interconnected system that even in 300 BC, he was able to infer was probably had to be true if we were going to be able to do things like complex reasoning. And then he knew that those blood vessels went back to the heart. So he reasoned that the heart would have to be the center of attention because of the computations the vessels were doing in the head. All right. So there's a current dogma, which is neurons do all the work, and blood vessels to the brain are important, but they're just providing pizza delivery. They're basically the feeder system, bringing nutrition from the body up to the brain. Now, that's useful. The brain does use 20% of all of our resources, even though it's only 2% of our weight usually. Kind of like AI right now again, where all of the gigantic server farms that are necessary for AI take a huge amount of energy. The brain does too, just to run the number of processes it has to run. It's really an energy hog. And so it makes sense that it's got a huge vasculature feeding it. This is one image of that. This is a human brain. And if we zoom in, I'll then show you a more detailed view. But this more detailed view is from a mouse's brain. And what we've done here is we've taken and made its blood vessels fluorescent, red, and then one kind of cell type, one kind of, it's called a neurogliaform cell, for anyone who would like to know. One kind of neuron type we've made cyan in color. Now, one thing, this is a bit of a spoiler alert, but I hope you can notice is just how much the neurons and the vessels are really nicely positioned to talk to each other. I've only labeled one kind of neuron in the brain, and there are thousands of kinds of neurons. And yet, even only labeling one type of neuron, you can see that it literally reaches out and contacts the blood vessels at a variety of places. So neurons are in a really good position to interact with the blood vessels. But the current view, more or less, is that blood vessels are just pipes. They're important for delivering stuff. Of course, we need them, but they're purely on the metabolism side of the kind of machines we need in our body. They're for delivering sugar, pizza, oxygen. They're not for doing something like changing the way we think about a situation because something new that we learn, or shifting our attention. If you have ever been at a crowded party and behind you, you hear someone say your name, and you might not even move your head, but instantly your attention goes to the person who said your name. That kind of transformation in our attention, blood vessels aren't supposed to have anything to do with it. They're thought to be static pipes. However, it's fairly well known that dilations, so an expansion in the size of a blood vessel, really nicely predicts the part of your brain that's actively doing information processing. So I'm showing you one movie of that from my laboratory. It's actually not a very good movie. This was one we took almost 20 years ago, but it's sort of dramatic, so I figured I'd use it as the example. You see how the vessel is moving wider like this? That's a dilation. Now, it turns out, using principles that Linus Pauling, who maybe should have gotten a third Nobel Prize, that Linus Pauling figured out, it's quite possible to measure those dilations without having to look at them, like the movie I just showed you. You can use them because as they have hemoglobin, as you change the amount of hemoglobin in a place in space, you can see a change in its magnetic field. And for that reason, if you use giant magnets and you tune them just right, you can actually see inside the human brain. It's really powerful. One of the signals that's most prominent to see is the change in dilations that are occurring because they change the amount of hemoglobin around. When we do this, and we have people do a very simple thing. This is actually a picture from my own PhD, where I tapped people on the finger and then simply had them answer very simple questions about it, or just sit there and passively perceive it the way I asked you to perceive the view from the porch. And it's a little bit different looking, but this is that same spot. It's kind of an inflated view of the brain to make it easier to visualize. Everywhere there's a little yellow spot was a place that dilations were occurring. So this is sort of the map of what your brain looks like when you're feeling something on your fingertips. These are the brain areas that are involved. And those are all mapped by following where those blood vessel movements are. These dilations are, put another way, really well correlated with information processing. It's how we've made the modern atlas of how the brain works. There are over 120,000, at my last count, papers using this method because it's an incredibly reliable way of mapping onto exactly where information is being processed. So here's that fuchsia colored region I pointed out to you before. That corresponds to right about here, where we saw responses to tapping on the finger. However, it's really interesting to note that these dilations don't seem to be very related to the metabolic need of ongoing activity in brain areas. Now, they're generally related. Where your brain is active, there's more need for sugar, or there's more need to clear out brain waste. But they're not very proportionally related. Every time people have tried to relate dilations to a metabolic need, it hasn't really worked. About every 20 years, there's a really prominent paper saying, surprise, it's less related than we thought. In fact, if you want to have a really high-profile paper, you can rediscover that with a new method. However, these dilations are really well correlated with where you're doing the thinking, which part of your brain is involved in the information processing. So the question then is, all right, we have dilations, and they're well localized to specific parts of the brain that are doing the information processing. As I'll update in a minute, that was our first criterion of this Turing test. What would we need an information processing system in our biology to do to help us understand information processing in the brain, or information processing that leads to things like the ability to imagine the porch? Well, neurons are actually in a great position to listen to blood vessels. So that this is a zoom in of the picture I showed you before. And you can see this blue neuron here, it literally reaches out and touches the blood vessel. Now, this happens to be a kind of blood vessel that's doing most of the dilating. It's called an arteriole, but it does most of the dilating that leads to that fMRI signal. And if it dilates, and if this cell, for example, has an ability to detect movement, which many neurons have, the ability to detect movement, it's an interesting fact, it would change the activity in this neuron, change its action potentials, and that could change the pattern of activity, and that could inform how information occurs. Now, this isn't the only kind of cell in the brain. I just chose one. It's called an NDNF cell, the blue ones, that sits close to a neuron. I'm showing you another picture here. This is a very recent picture from my laboratory, where this in gray is a very similar blood vessel to the one I've shown you here in red. But now here, these magenta colored dots are parts of the axons of dopamine cells. Now, dopamine is tied for first the most studied molecule, maybe in the world, but certainly in the brain, in topics of studying the brain. And it is because dopamine is the best predictor of what's important to you. If a lot of dopamine is present, you're more likely to complete an action. If a lot of dopamine is present, you're more likely to be able to learn from a just completed action. So you can figure out the right ensemble to recruit later in your information brain systems. And it's been known for years that dopamine is in a perfect position next to blood vessels. This has been known for at least 30 years, if not longer. And it's always been a puzzle why, but if I can use a demo here, these dopamine axons literally wrap around blood vessels. It's like an ivy growing up a garden post. It's amazing. When you see this, it's beautiful to see in brain's anatomy, these dopamine axons wrapping around. Now, they're literally so close that at times you can't fully resolve the boundary between the dopamine axon and a blood vessel. So the axons that have the ability to release to the brain dopamine, the molecule that is maybe most important for behavioral relevance, coding it, driving it, are so close to blood vessels. Here's an example where it's about 1 to 2 microns away from a blood vessel. And actually, they even get much closer than that. This isn't the best example of their proximity. They're in a perfect position to potentially sense something like a vessel dilating. And if you notice, I'll just put arrows here so it's really clear. All 3 of these different NDNF cells, another type just to switch around a little bit, are in a perfect position to listen to this vessel. I'm not saying that they do listen to it. I'm just saying that they're in the perfect position to listen if the opportunity arose. All right. So despite the current dogma, but in the beginnings of agreement with Aristotle, blood vessels are in exactly the right position in their activity patterns and in their dynamics of dilation, of moving, to correspond to where we know the information processing is happening that underlies our ability to do different kinds of things, like perceive our hand, or a different part of the brain is for, seeing our hand. All right. But dilations aren't as cool as action potentials. Action potentials are like I showed in the movie. It's like an electrical lightning bolt that comes down, and then a magical calcium thing happens, and there's a chemical release that then turns into electricity again. But recent discoveries in my lab, and actually in other labs, but some of it's just in our lab, and much of what I'm showing you now has never been published yet, so you're getting a first cut of it, actually have action potential-like events. They have calcium spikes. So here's one movie that shows examples of that. Now, this is a very sped up movie. It's going about 20 times as fast as it happened in real life. What you're looking at here is everything red is a blood vessel that we've made red by putting a fluorescent dye in the walls of the blood, in the tube of the blood vessel. And then in green, every time you see a flash, That's a calcium event in that blood vessel that's a lot like the calcium events that were happening in those neurons I showed you at the outset. You can see in this part of our picture, this is actually a dilation occurring. And actually, for this kind of vessel, the calcium events that happen just before it dilates. It's quite cool. But you can also see signals propagating here. See that wave? It literally comes down and then pauses right where my cursor is, and then goes on further. It's almost like it's stopping to think for a moment, and then it propagates the information. Now, this is, I think, kind of a famous, not famous, but important movie. This is the first movie of these calcium spikes ever taken in an awake being. It was an awake mouse that was running on a wheel and having fun and getting rewards and stuff, but it wasn't doing anything in particular. It wasn't doing a task or anything. And we make them very happy doing that, so we can take pictures of their brain while they're doing that kind of thing. Now, the next movie I'm going to show you is what happens if we actually stimulate those dopamine axons and then look at what happens in blood vessels. Now, the way we do that is a trick called optogenetics. The simple version is we take the same sort of molecules that are in your eye, opsins, that translate light into electrical activity, and we artificially put them into dopamine axons, so that now when we shine light, a dopamine axon will have an event a lot like an action potential. So what I'm going to show you is a movie, and when you see on the bottom of the screen a red bar appear, that's when we shined light, and that's when the magenta axons will be having an event a lot like an action potential, which means they're probably releasing dopamine. We're actually doing research on that right now. Really talented undergrad in the lab is actually doing a great project on that topic of how dopamine release happens in this case. In any event, what I hope you can see is right when this red bar turns on, I didn't mean to get in the way of it there. Right when the red bar turns on now, right here, you see red traveling down here. Now, in this case, the red moving here is one of those calcium events moving through the blood vessel. This is a blood vessel here, and that's red. But that's the blood vessel responding quickly with a calcium event to a signal that we drove in this dopamine axon. In other words, it's having a response a lot like a neuron that received a signal from another neuron and turned it into an action potential would have, even though these are blood vessels. So not only do they have the dynamics of dilations, we're also discovering that they have exactly the kind of electrical and calcium dynamics. Other labs are too, but not that many. This is a pretty unique area of research, actually, that they actually act like action potentials in the brain. So it looks like, we think, that there are phenomena that are fast enough in blood vessels to be able to potentially participate in the process of representation that would be necessary as a biological computer. And if you look at this image here, I hope it's easy to appreciate just how dense the blood vessels are in the brain. I hope you could appreciate that from the picture before. They're so dense that there's no neuron in the brain that's more than 30 microns at most from a blood vessel. It's exactly what Aristotle saw. He saw this really dense network in the brain and said, oh, that's where the information processing, that passes my Turing test, even though that Turing test hadn't been invented. I think Aristotle actually said that in ancient Greek, but whatever. It's a really dense network. I'm also just showing you one little snapshot of that network, not the full depth of it. There's a lot more richness to it. So they're fast enough. And if these calcium events are really independently occurring and traveling and talking to each other, they probably have the spatial precision to do a lot of the representation we'd need to do things like imagine, perceive, and act. Okay, but what happens when those calcium spikes happen? I mean, is this just a parlor trick? Is it just kind of a cool accident that they're doing that? Well, there are a couple different ideas. One idea is that the calcium events might actually help dilations to happen. If dilations then moved a mechanosensor on a nearby neuron, that'd be one way communication could happen back and forth between a vascular and a neural network. We've recently found something else, which is that we have strong data, but we're getting more of it to make sure we really understand what's going on, showing that calcium spikes create what we call plume events. And now that I've told you, I think this makes 150 people who have heard the term. So plume events are moments when another kind of dynamic happens. So it's not a dilation, it's not a calcium event, but just like an action potential has electrical activity, then a calcium signal, then chemical signaling, when we drive vessels in a way that creates those calcium events, we get another kind of chemical signal. Maybe the most important one you could have as a brain that's trying to make a calculation and trying to decide whether or not one should risk leaving one's burrow and leaving one's pups behind to go get a water, to go get a water source. That is all of the information in our body that we need to know about body state, that we need to know to best make choices to support our survival. Information processing in the brain, it's easy to forget as we sit and read our cell phones and play games and do things like walk down the hall unimpeded to go to the water fountain or to grab our water bottle. It's easy to forget that this thing all evolved to live in a very competitive environment, one where if there are bad decisions made about actions relative to what's actually needed by our body, it can be a life or death mistake. So what I'm about to show you is a movie that's very similar to the one I just showed you. But instead of measuring calcium in the blood vessels, we were actually measuring whether or not the fluorescent dye that's in the blood vessel would come out into the brain. And when you see the red bar at the bottom, that's when we turn the light on the dopamine axons. So there's the red bar. And all of this pseudo color you're starting to see now, the red, green, yellow, is actually signals being transmitted, chemicals being transmitted out of the blood vessel and into the nearby neural space. What I'm showing you is the same experiment, but out of 49 times we tried it, I'm just showing you examples of what happened on 2 different trials. And what hopefully you can appreciate, I'll put some arrows in to guide your vision, is how there's right by those white arrows at the bottom, there are distinct regions where this signal is leaving the blood vessel. There's a very specific spot where it's happening. So it's not a highly dispersed signal. It's actually precise in where it emerges. There's another region that creates plume events here on the side of the vessel. Now notice how close these neurons are to exactly that spot. This is a different data point than this one, but if this plume event is happening here, the one I'm showing you there, it would actually contact literally, and the chemicals would literally come in contact with that neuron. And these chemicals are the goods. This is all of the body signals you have that tell you whether you should be hungry, thirsty, have a maternal instinct, energized, not energized. All of the different body signals that control our behavior rightly in the wild, at least, that tell us what is our actual need on a whole bunch of timescales. But in the moment, that's what comes out into the brain when we do the same thing that creates those calcium events. So very much like an action potential in neurons, we think we're finding very similar chemical signaling events, but much richer because it's all of the signals from the body that could come in blood vessels. There's a calcium event and then a chemical release that could be signaling, for example, to nearby dopamine axons. They could call to the vessel and then hear what the vessel has to say about the body state, or to nearby NDNF neurons, which are really cool cells that we're very sure play some role in controlling our selection of ensembles. Oh, and those are the arrows again. So in that rich example I used at the very beginning, where the small mouse came out and it had to decide to go to the water source. The mouse has a lot of things it has to do. It has to choose whether or not to go there. It's got to persist even if it hears a hawk screech. And then if it succeeds in getting the water and getting home, it's got to learn from that event. So next time it can find the right brain ensembles. Now, we've studied this in the lab in a really simplified way. To understand these sort of actions and whether or not they're related to blood vessel dynamics for signaling, the kind of signals that help it pass the Turing test for computation in the brain, we've used a classic Pavlovian task. So as you may remember, Pavlov's dogs, they would play a tone, and then they'd offer food, and after a while, the dog would salivate to the tone. We've done the same thing, only we've been just giving really gentle stimuli to the whiskers of a mouse who's having fun running on a running wheel while we put a microscope lens on its brain. And we give that sensory cue. We just flick its whiskers, its vibrissae, for 2 seconds, and then we give it a reward. And we always do it this way. And that allows us to look at, would the vasculature have a response to the cue or to the delay period or to the reward, these key parts of what it means to have a behavioral perceptual action. Now, this is a movie, one of the movies I showed you before. To quantify this kind of movie, what we usually do is create a computer simulation of the walls of the blood vessel. And then we ask, where do we see a significant change in the fluorescence that's consistent in space and time? And we see things like this. So this is a colored rendering as a static image, not as a nice movie like that, of one plume event, one moment when some dynamic in the blood vessel, we think it's a calcium event, creates a moment when the vessel can then send a chemical signal to nearby neurons. Now, this particular event, I'll tell you more about in a second, was one of the ones that we see when animals are performing this task. We also see this green event here. And what I'm showing you in the box is actually this event, but how it occurs in time, in about real time. This is about the speed at which all of my movies, except that one really fast one, are shown at the speed at which they're happening in the brain. This is how a plume event occurs when it's triggered. So it's a cartoon of it. It's not the live movie, as I'm showing you on the left. But nevertheless, I hope it gives you a sense of the dynamics of this chemical signaling in the brain. All right. So now we're getting to the exciting summary slide. Makes me excited, at least. I hope it's making sense to you all. Does the vasculature of the brain, as Aristotle thought, pass the Turing test of maybe being a biological system that could allow computation in the brain? So it absolutely passes the first part. Vascular dynamics, in one example, the case of dilations, do occur in the parts of the brain that are known to be involved in specific tasks. In other words, they're generally in the right place at the right time. Okay, that's an entrance criteria for what could be a biological computing system that contributes to imagining raindrops on a porch. Are they fast enough? Well, I've sped this up, so no illusions there, but these signals are traveling pretty close to the speed they'd need to. We think there's actually a whole invisible world of fast lightning bolt-like signals, like the ones that trigger the calcium entry in the axon. And a really talented postdoc in the lab, Kevin Turner, is actually developing the methods for visualizing that right now. But do they have enough spatial precision? This is all within a few microns, maybe tens of microns. It's close to the spatial precision of neurons. It may not quite be as spatially precise, and it may be that the role of blood vessels isn't the full memory computer, but it helps you do things like shift your attention, so it kind of generally organizes behavior. Either way, the timing is close. We just don't know yet. And can it send signals that would interact with and drive nearby information networks? Well, they can. It's sending chemical signals that we know interact. It's sending dilations that can interact. And I didn't mention it before, but these calcium events probably should be able to trigger blood vessels to release other chemicals from the walls themselves. All right. So what are the implications of the embodied brain I'm showing you? And this is the last slide I'll show. Well, hopefully one implication that you would take from this is I think it helps to blur maybe not blur, link, that's a better word I use there, the body and brain. It certainly brings an appreciation that, you know, we eat with our body and we move around with our body, but we think with our brain, and the whole illusion is just up there in our neurons of our brain. What it means is that the systems that are entirely continuous between what is metabolic supply, oxygen supply, and the abstract system of neurons, it may be that the body is doing a lot of the computing that we typically think of as only what's going on in the neurons. Now, it doesn't mean that neurons aren't active. Neurons matter. They're super important. They're what I've devoted most of my time to studying, and I still think I didn't make a bad choice. They're why I'm employed. But it does beg the question of whether or not we should have named my field neuroscience. It's a field named after a certain cell type, the neuron. And that's a mistake if we want to understand that other body systems that are highly dynamic and pass the Turing test might be part of how we process information to create our perceptions. This is what we call an embodied brain. Now, what are the other implications? Well, as an extension of that implication, it really interestingly poses questions about health and how our brain works. So right now, we think that vascular disease can be bad for the brain and for thinking because we're damaging information, energy supply. Maybe we're also damaging part of the computer. Maybe the reason that memory problems emerge in Alzheimer's is the really well-known and well-studied changes in the vasculature in Alzheimer's aren't just a loss of energy supply or waste removal. They may also be a change in the actual computer that holds the memory. And I guess the last implication that I think is really exciting to think about, and that I don't know that I have the right answer to, is as we think of problems of how it is that we have awareness, how it is that we perceive states that are far more complicated than our own narcissistic consciousness, our first-person perspective. Maybe we're part of more global consciousness. How it certainly is bringing us in closer alignment to that. You know, holding hands, having a hug, interacting in these ways. It may be that these are far more related to our actual computational structures in the brain than we appreciated. Certainly that intuitions, gut senses, feeling like, all the metaphors we use are metaphors we take from the body when we're saying somehow we know something and it's even more complicated than I know how to express, but I have a good idea what to do. Maybe the body is actually physically contributing to that sensation. So that is all the slides I have today, and I'm super eager to talk with folks.
Okay, thanks so much, Chris. That was really interesting. So as a respondent to kick off our commentary and question and answer, we have M. Catherine Bennett. She's a neurobiologist who recently retired from NIH, where she served most recently as a scientific review officer. She recalls that her first job was as a co-op student in the NASA-affiliated Navy lab back in the 1970s. Kate has worked for 50 years in various fields related to neurobiology, with more than 60 published articles to her credit. She is a charter member of the board of the Institute for Science and Judaism and served on the board of the Haberman Institute for Jewish Studies. Kate brings to this discussion a lifelong interest in fundamental questions about the origins and meaning of human existence viewed through the lens of physics, cosmology, neurobiology, and religion. So, Kate, can you have some responses?
Okay. Wow, Chris, that was great. It's a tough act to follow, especially because as we've corresponded, this really is not precisely my area of expertise, but I was avidly taking notes, and you'll have to forgive me because some of the memory joggers I have are written down, and I have questions that range from highly specific, just how does this work thing, to global kinds of questions. And if you'll bear with me, and we'll try to dissect those together. So as I was reading your, in several places, you were talking about the dopamine modulation of the, your hypothesis is that it modulates the vasodilation in these microvessels. These can't be the major, I mean, I don't know, the vessels you were showing seem to be the ones that are in the microvasculature, because those would be in the best position to influence individual neural activity. So what the first question that occurred to me is, you said in one of your blurbs that there's a chemical signal that dopamine releases that activates this vasodilation. And I'm just wondering, pragmatically speaking, have you identified that? Or is this just a direct dopamine agonist function on the, I did get the chemical signal from your blurb.
Yeah, sure. It's a question. You're right on target. It's one that is very consuming to us, and we don't really know the answer to. I can say a few things, I guess. One thing we know is that when we do something really fake and actually just pour a form of dopamine straight onto the brain, we see the blood-brain barrier open up, and we see chemicals that are in the vasculature come out into the brain. It's actually a really remarkable effect. It's a really big effect, almost surprisingly big, enough that we don't totally trust it yet. So
can I interject a question here? I hadn't put this together yet. Are you suggesting that the vasodilation is actually simultaneously opening the blood-brain barrier?
I'm realizing I explained something really badly, which I'm good at doing.
No, I'm sure it's my ignorance, but
I'm world class at it. Let me just see if I can do something here. Let me share my whiteboard for a second. All right. Can you all see that can you see the whiteboard? Is that
Yeah.
the screen?
Yes.
Yeah,
So,
good.
all right, thank you. So if you have a blood vessel here, let's say, and these, to give you a sense of size, are about 10 microns across. So they're actually thinner, or as small as 5 microns. So they're like half the size of a neuron cell body, depending on what neuron you look at, of course, as you know, Kate. And there are 3 kinds of dynamics in blood vessels that I was talking about. One is that they open up and vasodilate in the parts of the brain that are actively processing. So if you're processing touch, you'll open up the touch maps. If you're seeing something in vision, your visual cortex will open up. There's a second kind that I showed you, and this is the kind that you all saw, more or less the first movie that I know of in an awake animal ever of. They can actually transmit, I'll use the chemical abbreviation, they can actually transmit these calcium signals up and down their vessels. So it doesn't mean that necessarily went with the vasodilation. Some people think it does. We definitely have lots of examples where it has nothing to do with vasodilation, and then sometimes it will, but it's not the same.
Calcium does a lot.
Calcium, anytime you want to like publish a paper, study calcium.
Yep.
There's this third thing that maybe I'll do it this way. In your blood vessels are a whole slew of signals from your body about should you be thirsty, should you be, all of the hormones, all of the metabolites, all of the protein signals, potentially even antibodies to some extent. There's all of those signals are carried in your blood. It's like a fire hose of information. It's an unbelievably rich source of information about what you need, where your body's at. All of those signals are carried around the blood to some extent. Not all of them, but many, many of them, as you know. And the new thing we've discovered is that you can have very specific, oh, shoot, I drew that wrong, is that you can have very specific moments when the blood-brain barrier, which is the barrier that usually keeps these signals from being transported, opens up. Now, these are what we call, these are the, now you have to see my handwriting again. These are what we call plume events.
Yes.
So that's a dynamic that is a chemical sensing or a chemical transmission of all those body signals that could go out into the brain in a way they don't usually, and potentially influence a nearby neuron. We'll name her N here. Let's say she's got some sensors, and at least one of her sensors is in the path of those chemicals being released. So it's by having the blood-brain barrier be dynamic, it's a way of getting a really brief, very hyperfocal in the brain snapshot of what's going on in the body. That's one kind of dynamic. The calcium events are another, and then dilations are another. There are three different ways in which blood vessels are dynamic. Now, the calcium events, we also think are related to voltage events, but that's not something we need to be complicated about. We think these calcium events do cause the plume events. The lightning bolt entry of calcium, just like in an action potential, just like in the most accepted form of signaling in the brain, we think that causes a chemical signaling event. We think the calcium event in the vessel causes a chemical signaling event, which is letting the brain get a snapshot of where the body is at. We think that's potentially a really important mechanism for being able to make the right decisions based on your body state, based on your real needs, not on whatever ideas are running around in your head. Does that make so? I really kind of blurred those in my explanation. I'm sorry about that.
No, I just want to say to the people who are listening, to really do justice to this topic and in a real Q&A and detailed, this would probably be a 3 to 4 hour discussion. So I'm going to try to stay on point more than I have, and maybe not open up as many of the micro questions as I had. But do you see these things, just quickly, do you see these things as sort of exogenous hormones that have cell-to-cell communication? Okay. Okay, so let me just, I wrote this down, so I want to just say it because I can read it faster than I can recapitulate it. So we know that fMRI and BOLD, these are two measures of brain imaging. One involves the B in BOLD stands for blood activity, so that we know that activity and blood flow correlate with neural activity. However, my first question was answered within your talk because I was looking at the standard model. Why isn't this evidence of simply reflecting that information processing is simply being provided on demand as the ATP is needed? And you actually addressed that. So I would say that a priori, I was a believer in the standard model of this. But I love to be shown contrarian hypotheses somehow, and I don't know how it happened to me. 4 or 5 times in my career on big things, I ended up being a salmon swimming upstream, and it's just the way the data came out. So I love contemplating alternative hypotheses. I'm going to skip over what I wrote about the Turing test. I will just say briefly that I'm not a huge fan of it. I think we've gone a little bit beyond that sort of simplistic way of looking at whether we're talking to a computer or a human. And let's see if I can simplify this. So you wrote down 3 things, 3 or 4 things that were implicit and needed for the Turing test. So activity should predict brain locations. It should be acting as fast as the phenomena that explains. It has to be precise enough to explain phenomena. Support details. So we need to move fast and precise. And it can send, and I added this, it can send signals to other networks, which is the connectome, which is how I would personally see these simultaneous actions that could appear to be at a local level, but they're actually distributed across all the, just for the human brain has somewhere between 85 and 100 billion neurons. That's just that neuron type cell. Every neuron makes, on average, 100 contacts with other neurons at its axonal site and 1,000 at its dendritic site. So combinatorially, you've got more than entities in the universe that we know. That's just, to me, a little interesting factoid that shows how we can put these things together and imagine, essentially, for our purposes, an infinite possibility of ways that these can interact. I added that something that you didn't mention, I'm sure that you meant to because you alluded to it later. We also need a memory system, because I just talked to my Alexa and I say, Alexa, I missed what you just said. She says, I have no way of going into the past. Okay, so we also need to add memory into that Turing test. Okay,
Great.
I was
Learning and memory, I think, is one of the most important things that would have to be shown of any biological system that really supported something like imagination or intelligence. I completely.
so when just a quick story, I was put into a hormones and behavior lab, and in one semester I decided that even if I solved what I considered the most central problem of hormones and behavior, that is the genetic hormonal identification of what makes gender differences, I was not going to find that to be a satisfying career. I thought learning and memory was where I belonged until I found out, and I was a student of James L. McGaugh, you might know him, very famous in the field, until I learned out that learned that we were simply looking at gain. We were simply looking at modulation. We were looking at how to make memory stronger or weaker. And I didn't want that. That's when I got interested in the information processing aspect. I promised myself I wouldn't go past 6:10, and I'm already 3 minutes past that. Very quickly, here's where my antenna went up. I said, every 20 years or so, a new paper is published saying that the vasodilation is not so correlated with metabolic demand, but is much more correlated with information processing. This is where you started pulling me in. So let me just jump over some of this and try to ask you some open-ended questions. My general question to you, and I think this is a huge question, you're certainly not going to be able to answer it in 15 minutes, but it's one of those, let's think about this. How do you envision this influencing decisions, and how does it create action? Because I studied I was pulled into the Parkinson's field because my model of Alzheimer's disease looked more like Parkinson's than it did like Alzheimer's. So I became very interested in studying extrapyramidal motor system, mostly the nigrostriatal dopamine system. And that's where I jumped in, and we probably have the most overlap of our fields in that area. I don't think neurons are irrelevant. I think they're extremely relevant. And I know you don't either. I'm saying that almost as a tiny provocative question that leads to the question of how do you see your mechanisms, your putative mechanisms of the vascular contribution interacting with these known networks we have? Because back in the 80s, I read a wonderful book that I think is still timely called The Organization of Action. It is a classic in the field, and it is absolutely beautiful, and it still is current. So we have systems that take in sensory information, sensory motor integration, we translate it to actions, we send it through the cerebellum, we send it to the striatum, we send it to the descending axons in the spinal cord, and that was, until your talk, my general idea of how we produce sensory motor integration. I'm stopping.
I got it. Let me ask everybody. This is fantastic, Kathy, and this is really really great to talk with you about it. So let me ask everybody to do an at-home experiment right now. So everyone take something like a pen. Don't uncap your pen. Take something like something that won't break you or something else. If you throw it and then throw it in the air and catch it, or just wave it around. Choose throw and catch or
Very uncoordinated.
well, it's making it all very dynamic. All right. Now, when you threw it in the air and caught it, what caught it? What caught the pen? Assuming you caught it. But
Very fast calculations that the cerebellum makes and the nigrostriatal system make that coordinate the position in space that your hand has to be.
I totally got you, and I probably agree. But the answer that you're giving, which I agree with, is a great way to think about it, is that the neurons did the work, but actually the hand did the catching, right? And it had to have muscles. I'm not saying anything, I'm saying a simple thing here. I'm not trying to say something. You had to have muscles work, but you don't think the muscles did the work. They were dynamic. There was a lot of things going on, but they were just kind of like in service of a commander.
Yeah.
The command. What if the blood vessels are treating the neurons that way? Like, yeah, you got to work with the neurons. They're the readout, but we're really in charge. And actually, we do the computation. We send it to the neurons and they implement. That's nice. And then the muscles follow the neurons. But really, the prime mover, which is hopefully a phrase we take the right way in this call, the prime mover is the blood vessels. Now, I actually don't think it works that way. I think all of these systems interact together. Actually, I think muscles are much smarter than we give them credit for. And I think neurons are a big part of it. They're the crown jewel of how we compute in the brain, but they're not the whole crown. They're not the whole, they're not the whole thing. And they only probably succeed, and we'll probably only get the problem right if we include all the systems that they're talking to, and they wouldn't get. So I think the way I personally, if this was Monty Python, have you seen The Holy Grail, the movie The Holy Grail?
Long time ago.
There's a bridge there, and if you answer the question wrong, you're thrown in the pit of despair or something, right? So if you put me on that bridge and ask me, well, what do you really think? So I got to be honest. I'm not being rhetorical. I think blood vessels are part of the computation, but I bet what they're part of is sort of helping choose the right set of neurons. Like you wouldn't make the right choice of the right set of neurons, but they're probably representational, somewhere more like what we might call a modulator, but a very specific modulator, and one that's very important and timely, and you wouldn't get it right without it. That's my guess, is that it's not memory. It's to your point about gain. It's not memory itself. It's like the brightness or the choice to go towards one thing or the other. In other words, you wouldn't get the right selection. You still need the neurons to be the selected thing, but you wouldn't get it right though without the blood vessels. And so that's part of the information. But it may not be a substitute computer at all. It's probably interacting and doing the unique things, like knowing about body state that blood vessels are great at, and then helping the neurons to see that part of the information. Does that make sense? So
it does, but I'm also realizing we're getting signals that we're monopolizing, and I apologize for my contribution to that. So we need to take questions.
Yeah, this has been a great discussion, but yeah, we'd like to get in a few questions from the viewers. So there were a couple here about, are there any thoughts, or can this be related to causes and cures for dementia? And someone mentioned MIT research.
Yeah.
Reducing dementia using 40 or 60 cycle per second signaling. Is this related to that?
For what it's worth, that therapy for Alzheimer's came from this paper, which was a Nature paper I published 15 years ago, 16 years ago.
Oh.
I was actually the outside thesis examiner of the student who originally did the work. What's being done there is, if you provide the brain with stimulation in what's called the gamma oscillation range, it's the name for it. It's a really interesting and mystical brain rhythm. If you've heard about EEG brain rhythms, like sleep rhythms, and It's the one that's sort of the attention or consciousness rhythm to some people. And to some people, it's the exhaust fumes of computation in the brain. It's like an accident that happens when your brain is doing the work. And we invented a method for controlling gamma so that you could actually ask that question, can it change behavior? And my colleague, my collaborator on that paper then reasoned that if you could drive, if things like doing meaningful tasks and playing brain games was good in Alzheimer's because it was recruiting those circuits, what if we just drove them directly with gamma? Like we went right to the kind of pattern of activity in the brain that was being created when you do something like a crossword puzzle or, paying attention to a topic. And I actually saw this colleague yesterday by chance. Her name is Dr. Liwei Tsai. And what Liwei found was really remarkable evidence that driving the brain at gamma in a variety of different ways, driving that pattern, either in through the eyes or directly activating it, using the same trick I was using on those dopamine axons, optogenetically. The both ways of doing it, you would actually, in mice that had symptoms of Alzheimer's, which is very different than the human disease, it's important to say, but in mice that had symptoms, you would actually see a relief of some of the symptoms, and you certainly see a relief of some of the changes that happen in the brain. They've been trying this in humans in clinical and preclinical studies, and I think there's evidence that it's working. There's certainly some evidence. I don't know how far along it's gotten, and I'm not really expert in it, but one way in which that touches on this work is there's a very specific cell type in the brain you can stimulate to get gamma. That's the trick we used in the paper I just showed you. It was great that it's a cool coincidence that someone asked that. And that cell type is the cell type that in my lab, we've discovered, it's internal, we haven't published it yet, is the closest to blood vessels. It literally sits so close to a blood vessel, we can't tell where the border is. Like dopamine cells are close, and those other blue cells I showed you are close. This cell type, which is called a fast spiking or parvalbumin fast spiking cell, literally sits so close that we can't see the membrane boundary between it. You could with some methods, but not with the kinds of images I was showing you before. They're really intimately interconnected with blood vessels. And so what I've always thought Liwei was doing, Dr. Tsai was doing in those studies, was by shaking up or driving those cells in gamma, she was actually causing the blood vessels to do beneficial things. I think it's just a really direct pathway to getting the blood vessels to do their work. So I do think that there's a lot of avenues to treatment in dementia. I certainly think that given that you can create beneficial blood system dynamics by doing small rewarding tasks, that the whole idea of playing brain games is a good one. I'm behind it. I'm hugely behind it, actually. I think it matters that it matter to you, but I'm a believer that it matters.
Okay, another question, maybe the last. What triggers vessel dilation? I'm thinking of ways where we use pressure on our bodies to calm our state, like swaddling a baby.
That's a cool thought. I do think that the question being asked is on to the really exciting place where it helps us to understand all these ways in which we intuitively understand that our intelligence and our emotional state are really closely tied to our bodies. And it feels like more than just I ate too much sugar. All of our metaphors about knowing things, almost all, and lots of them come from body metaphors, right? I feel that, or my heart's pounding, but we actually mean I'm excited. I do think there's a direct connection. It's a lot of things can cause the dilations is a short answer. And that probably makes sense because just like neurons would want to talk to a lot of different other kinds of neurons, there should be a lot of different kinds of things that can converse with the vasculature. But I think you're exactly on the right track, that things like, and in autism, often in autism, that kind of swaddling can be really helpful to kids. Not always, but often that sort of pressure sense can be really useful. I do think that's connected. I don't know exactly, I wouldn't want to overstate it either, though. I don't know
Mhm.
exactly how, but I think that's the kind of connection that this line of thinking, if it's right, it begs that kind of connection. Yeah.
Okay, well, thank you. And there's a few more questions. I think we'll be able to pass them on to you.